Hi everyone, so today we're going to do the Canvas Kit follow along video. What that means is that I will go through the entire Canvas Kit experiment in real time with you. I'll mention some things to look out for and really just show you how it works so that when you're ready to do your own experiment, you're confident that you'll succeed. Or if you don't have the chance to do your own experiment but are still interested to learn how agar art or bacterial paintings are made, you can see what the process is and how it works for creating art with bacteria. So here I've laid out all of my supplies. What I'll do first is I'll put on my gloves. So bear with me a second. Alright, and now that I've got my gloves on, next step is to clean up my surface. So I'll move my supplies aside, give it a wipe. Right now I'm using 70% ISO, isopropyl alcohol, which means that this will disinfect my area. If you have a bleach cleaner, that's fine too. You'll also notice I am working on a sort of metallic surface. So what's great about this type of surface is that it's non-porous, so it's great to clean up. If I spill anything, then it won't, absorb into the surface. So we really recommend you use this type of surface. So I'll just wet my paper towel and give it a quick wipe. Okay, I'll also do over here. All right, perfect. So I'll show you the supplies I have. So I have the Canvas Kit, which is all my experimental supplies. So it's got that bacteria, it's got the Petri dish, it's got everything I need to make the art. Then I've got my cleaner. I'll just put this to the side. I've got paper towels, put this to the side. I've got the Zero to Genetic Engineering Hero book, because what we're actually doing is experiment from chapter 3. So if you have the book at home, you can follow along. The steps are all there. And then we also have a few of the going deepers and the pro tips that I'll mention during this video. I'm just going to put this book aside for now. The other thing that I have is just some scissors to help me get into the kit, and then a plastic container, and this is like a recycled container, and I'm just going to put my inactivation bag in here. Then I've got a Sharpie, pretty important, and I've got some coloring pencils, and these will really be used tomorrow, so I'll put them aside as well. I've got a little UV flashlight. So this is actually a black light, and it's going to help us see some of the colors, because some of the paint in the kit, the bacteria paint, it does fluoresce under black light. So I'll put this to the side too, and I will get into my kit. So you'll notice here on the side that the kit will have a best by date. So for me, that is July 2022, which means that if I keep my kit in the refrigerator until then, my bacteria will still be alive by July 2022. So make sure you refrigerate your kit when you receive it. Make sure that you also take note of that expiration date, because you want to use it beforehand. The issue, if you were to use it afterwards, would be that your bacteria might no longer be alive, which will make it difficult to do your art. So any debris, any garbage like this, I can put in the regular garbage because it's not touching bacteria. So the first thing I'll take out is my inactivation bag. So this bag has instruction on how to actually inactivate your experiment sample, which means that you don't need to actually, do any burning of samples. If you're familiar with other types of inactivation, so this is really just a bleach inactivation, and it will kill everything that we have. So what I'm doing here is I'm just wrapping my bag around this container, and that's just going to make it easier for me to just throw away my supplies, right? So it's like a little garbage can. Put this aside. Then we can look at our kit material. So in here I've got some loops. I've got my water, so this is sterile water, and the reason we use sterile water is actually because in your tap water there might already be some microorganisms living in there, and there's some salt and minerals, so we don't really want that inside our experiment, so that's why we use this sterile water. Then I've got my agar to make my Petri dish. I got my Petri dishes. I got my painting tools, so these are sterile paintbrushes. You'll notice they're actually Q-tips and toothpicks, but these have been sterilized, so we know there's no living organism on these, and I can use them to do my painting tomorrow. Then I've got my stencils, and we'll talk about those tomorrow as well. And I've got my bacteria paint, so I've got 3 colors. I've got the cyan, which is the one that fluoresces under black light. I got magenta, which is visible under regular light and also fluoresces. And then I've got purple, which does not fluoresce, is only visible under regular lighting. So I'll put the supplies that I don't need right now back into my bag. So that's my stencils, that's my paintbrushes, and some of my loops. I'll need 3 yellow loops, so I'll just put the rest in here just to keep a clean space. So the first thing we do today is actually make our Petri dishes, and the reason we make our Petri dishes first is that these are going to be both the surface for the paint to grow on to make our painting palette, but it's also going to be what we make our art on, so there are canvases. So first things first, we make those. And what we're going to do is I'm going to open the baggie so that I can identify them. I'm going to use my Sharpie marker, so any permanent marker will do. And on the underside of one of the Petri dish, I'm actually going to make divide it in the number of colors I have, and this will be my painting palette. And the reason we do a painting palette here is just like in regular paint, when you use, you know, acrylics or oil-based paint, a lot of artists will use a painting palette, a board that has all of their colors on it, and so we're doing the same thing here, just so we have enough bacteria paint to do all of our art pieces. So I'm also going to write my initials on this Petri dish, because if I was in a classroom or in a shared lab space or even at home, I'm going to identify that these Petri dishes are mine, I know what's growing on them, and if anyone has a question about it, they can come to me. So my initials on all of these. Okay, and then one of them, like I said, I'm going to divide. So I have 3 colors, right? Purple, magenta, and cyan. So I'm going to divide it in 3 sections. I'm going to try to make them equal, but you can see that they're not so equal, but that doesn't matter. I'm going to do a paint streak here, a paint streak here, and a paint streak here. If I want, I can say that I'm going to do the cyan here, I'm going to do the magenta here, and I'm going to do the purple there. Okay, and you'll notice that I did write this on the underside of the Petri dish, and that's because when you pour your Petri dish, your agar is actually going to go in this section of it. So the markings, I want my markings to be on the section where the agar is, because the lids could be lost, they could be interchanged, but of course where the agar is, that's always going to stay with the marking. So I'll just move these back up. So now I need to fill them with agar. So to do that, I'm actually going to take my sterile water bottle and I'm going to microwave it. The reason we microwave it is that we're going to mix in this powder called agar, and agar is very similar to jello. If you've ever made jello or gelatin, it needs hot boiling water to melt. So I have my microwave here. So before I microwave, what I want to do is actually loosen the lid. like this. And you might need a parent's help to open the lids because they're pretty tight. But by loosening the lid, I make sure that as the water boils, there's no pressure buildup in this bottle. And because if there is, it could actually explode, so it could burst. So I really don't want that to happen, so I just loosen it a little bit. I put it in the microwave. And then I'm going to do 50 seconds. I don't know if you can see. And really my purpose here is to see the water boil. So I want to see rolling bubbles. And this is similar to if you've ever boiled water for spaghetti or rice, or if you've seen your parent do that. You kind of want to have those big bubbles where you can see that it's actually boiling, and that's really important. So you cannot really overboil your water, but you can definitely underboil it. So I want to make sure that my agar dissolves, because if my agar doesn't dissolve, I don't get any Petri dishes, and without Petri dishes, I can't go on with my experiment. So I'm just going to pay attention to what's going on. I think it's been, yeah, it's going to beep soon, and I think through the door I can actually see some boiling. So I'm going to try. So I don't know if you can see that it was at a rolling boil. So I'm just going to microwave it another few seconds, so hopefully you can see it on camera. Okay, I can see it. So I'm going to open it and show you really quickly. So I saw some bubbles. I don't know if you can see them. So it doesn't matter if it stops boiling. We need to see those bubbles happen in the microwave. So what I'll do now is I'll add my agar. Okay, so this is the tube of agar, and it's pre-measured for you. So all you need to do is really uncap it and pour the powder in. Now there might be a little bit of powder that stays on the lip, and that's okay. It's been like measured for, we know this is going to happen, so it's going to be measured for that. So what I'm going to do with this is actually put it in my inactivation. I'm going to put my lid back on, but I'm not going to do it fully. I'm not going to tighten it fully. I'm just going to give it a few swirls like this. So now be careful, because this is actually boiling water, so it's really hot. So what I'm going to do now, I'm going to re-microwave it for a few seconds, because I want to make sure that it's perfectly boiled. So I'm going to put it back in the microwave, and I'm going to do it in 4 seconds increment, and what I want to see is some foam on the top. So I'm just going to pay attention. I hope you can see in the video. But now I can see bubble and foam. And I'm going to mix it again. If you're not sure, this is really the step where you can make sure that it's been boiled. So you can go for another 4 seconds. So why don't we do that? Okay, I want to make sure that I don't leave it in for more than 4 seconds, 5 seconds at a time, because I don't want it to spill over. All right, so now I'm pretty confident that this agar is dissolving. So what I do now is I mix it for about 30 seconds, bearing in mind that it's still very hot, so you kind of want to hold it by the top and just gently swirl it, right? You don't want to shake it up and down, especially because your lid is not tight. And I'm going to get my antibiotic ready. So this antibiotic is just going to go into our agar, and the point of this is to make sure that only the bacteria that we want actually grows on the Petri dish. So in your book you can learn a little bit more about antibiotic selection, but really all we need to know right now is that the bacterial paints that we have can survive in the presence of this antibiotic, but the rest of the bacteria cannot, right? So naturally occurring bacteria cannot, which makes our experiment a little bit more precise. So I'm just gonna check in here. It seems pretty clear. And then I'm just gonna drop this in. Okay, this is done with. So this one I'm gonna put in my inactivation as well. And now I'm gonna put the lid on. I'm going to tighten it and just keep swirling until I see my antibiotic pill dissolve. So you'll notice I'm not shaking up and down, because I don't want my agar to become very bubbly, right? I don't want a foam or a lot of air to get into it, because when I pour my Petri dish, if I do that, they're actually going to have bubbles inside of them. And you kind of want to try and get a nice flat surface when you're pouring Petri dishes. All right, so this can take 45 seconds, a minute, and what you want to be seeing is actually, you can keep opening it up and seeing if that pill has melted. So I can see some debris at the bottom right here. It's white. That means it's not fully mixed in. So I close it back up and I keep mixing. Oh, so I'm mixing it a little bit vigorously, so I got some agar on my glove. So that's not really a problem. It was just a little hot, so I noticed. And then I'm also doing a few drops on the table, so that just means I'm gonna have to wipe my experiment space after, which I should do anyways. So let's have a second look and see. All right, so I'm seeing an agar liquid that is very clear. I'm not seeing any debris from my pill, so I think I'm ready to pour my plates. Before that, I'm just gonna wipe my table a little bit. All right, I have 4 Petri dishes, and what I want to do is fill the bottom half of these Petri dishes. If you have the Petri dish with the ring, sort of the ring on top, the side with the ring is the bottom. The bottom of a Petri dish is always the side that has a smaller diameter, right? So the lid fits on top of it. All right, so I don't want to be spending too much time talking and letting my agar solidify in the bottle, because I actually want it to solidify in my Petri dishes. So I'm just going to go ahead and pour this. So I'm going to open the bottle. I don't want to be speaking when I'm pouring, because I don't want to introduce any environmental bacteria, anything from, you know, my mouth or anything like that, or my breathing into the Petri dish. So I'm just going to pour them quickly. I'm going to try and keep the lids on as much as I can. All right, so you'll see I still have some agar left in my bottle. That's okay. There's a little bit more in the bottle than you need. So you'll notice I filled my Petri dishes half full, about to where the star or the ring is, right? And you'll notice that when I was doing it, I kind of left the lids half on. And so if you're in a room with nice clean air, so for example, you've got HEPA filters running, or you know that there's no environmental contamination or mold or anything like that in the air, you can leave them open like that, and they'll dry faster. And by dry, I mean solidify faster. So depending on your climate, your Petri dish will dry or solidify in anywhere from 7 minutes to, you know, it could be 40 minutes. It really depends how humid the air is. Where I am right now, the air is pretty dry, so usually our Petri dishes are solid in 10 minutes. So what I'm going to do is I'm going to leave them. I'm going to put the lids back on, because there's no rush for me to continue this experiment. If you are in a time crunch in the classroom, you can leave the lids off. So I'm just going to do this, and I'll get back to you when the Petri dish are solidified. I don't need to wait next to them. I can just leave them on a table, as long as I don't have any younger siblings or pets that can get at them. I'm good to leave them right here. So I'm going to take off my gloves, stop filming, and I'll see you in about 10 minutes. Okay, so we're back to check on our Petri dishes to see if they are solid. So what I'll do is I will open one of them, and I can see that there's no movement. A good test to see if they're solid is just to tap on the side, and if nothing jiggles, then you are good to go. So you can see it's absolutely 100% solid. You'll notice there is some moisture on top of it. That's because they dried with the lids on, so I will just leave them open a little bit. I don't really want any moisture on top of the agar because when I streak my bacteria, it will make it slide all over the place, so I kind of want to try and avoid that. So I'll leave them open a little bit while I get ready for my next step. And the Petri dish I will need is the one with the 3 sections. So when these are a little bit more dry, we'll put them back into the baggie and put them in the fridge. So in the meantime, what I did do was get my playground ready. So I have the home playground here and the large classroom playground here. The classroom playground is actually, if you have one at home, it will likely be red topped, but basically it's just slightly bigger. So in this one you can incubate 8 Petri dishes at once. So if you're using a classroom kit, you would have 8 painting pallets, so they would all go inside this incubator. In the home playground, you can put 2 Petri dishes to incubate at once. So I'm just going to put my 1 painting palette in there. And if you don't have playground, but you do have your own incubator, you kind of want to start set it to 37 degrees Celsius. So that's what mine is set to. Well, it's getting to temperature. I started it just a little while ago. And if you don't have an incubator at all, that's also fine for the canvas kit because the bacteria will grow at room temperature. The closer you can get to 37 degrees Celsius, the faster they will grow and the brighter the color will be. So if you don't have an incubator, what you're going to do is you're going to put the Petri dish inside a plastic bag. So you can get a Ziploc from home or something like that, and put it in, and then you're going to want to leave that in the hottest area you can in your house that doesn't go over 37 degrees Celsius. So you might be thinking, all right, I'll put it right next to a vent, but make sure that the air that comes from the vent is not too hot because that could actually dry out the plates and kill your bacteria. So you can also make your own incubator using an incandescent light bulb and a few plastic containers, and we do have a tutorial for that, and that will be linked in the comments. So if you want to make your own incubator, you're more than welcome to. It's quite easy. It's like a 5-10 minute process. Okay, so while we were talking, our Petri dish were drying. We have this one. I think it's mostly ready to go. So what I'm going to do is I'm going to take 3 of my yellow loops. I don't need this blue one. And so these loops are used for streaking bacteria or transferring liquid. The yellow ones are 10 microliters of liquid if you fill up the hole here at the end of the loop. But in this case, we don't really worry about how much liquid they can transfer because we're just going to use them as a streaking tool. So we're going to get, and I can touch these with the end of this while it's still wrapped up, but I wouldn't touch the loop when it's unwrapped. But I just want to show you. So we're going to use the end of this loop here, and we're going to dip it into the bacteria, and then we're going to streak with it. So we're just going to spread the bacteria onto the agar surface. So that's what the purpose of this is. And I have 3 because I have 3 color, just like I have 3 section. If you're using different paints, or if you have other bacteria paint from an engineering kit, for example, then you could separate your Petri dish into 4 section, 5 section, and you would just use the same amount of loop as the amount of colors you have. So I will open my bacteria tubes, my bacteria bag tube of tubes, and I will start with one color. I'll start with the magenta. Now you can kind of see that there is bacteria here, right? So what I want to do is find my area. So the area with the M is where I said I would put my magenta bacteria, and I'm going to take one of my loops. So again, you don't want to touch this side with your fingers or even with your gloves or any surface once you open it. So I'm going to open it from this side. A trick is to just push through, right, so that your loop can come out. And I'm going to also open my tube. Now, what's in this tube is actually another form of agar. It's a little bit softer than what you have in your Petri dish. And then there are bacteria that have grown in here, and that's how they stay alive. So if you can see a little bit of pink there. And what I want to do is I just want to put my loop in. And so it's going to be a little squishy, right? Because it's a soft agar. And I just need to dip once, and then I take my loop out. You'll see there's some agar that wants to come with me. I'm going to try and avoid that, but if some does get on my Petri dish, it's not a big deal. You can see my loop is wet, and there is a little bit of a shine to it. There's a little bit of agar on there. So I know that I've actually dipped inside the bacteria. And so now what I do is just a zigzag. So I'm going to do just a zigzag here. And what that does is it actually just spreads bacteria all around this section of my Petri dish, so that when I incubate it, a lot of the magenta bacteria will grow. So now that I've done this, I'm going to put my loop in my inactivation container, and I can actually keep this tube of bacteria in case my experiment doesn't work out, in case they don't grow, or if I want to do more art later on, or if I'm in a group. I'm just going to put my lid back on, or I would pass it to my colleague if they were also doing a painting palette, and I'm going to put it back in my bag. I'm going to take a second loop, and I'm going to take a second color. So I'm making sure not to touch any surface with my open loop. And then I've got my purple bacteria. Again, you can see sort of a purplish in there. I go in, and my loop doesn't go all the way through, right? Because it's bigger than the end. But that's okay. I just need to dip it once. I take it out. Now you can see that there is some agar. I can see some purple, right? That's fine. It doesn't matter. I can just go into my purple section, which was that big one here, and streak. So I just want to do a zigzag. I just kind of want to cover this area. Okay, so you'll see here, I'm putting my loop in the inactivation, but you'll see here that there's a big blob of that agar, right? Well, that actually doesn't matter because this is my painting palette. It's not my art, right? This is the paint I will use to make my art. So if there's a little blob on there, it doesn't matter. I did cap my purple bacteria, and I'm going to put it back in my baggie. And now I'm going to do the last one. So last loop, again, I push it through here. And so this bacteria is actually the cyan. So this one, it appears white under regular light, and then it glows cyan. I'm just going to gently grab my UV light, making sure that my loop doesn't touch anything. So can you see how this is cyan, right? So see nothing, or a whitish glow, and then you can definitely see that it's cyan, greeny blue color. So again, I'm going to go in. It's a little jelly. I take my loop out. I cap my tube. And then I streak in my last section. So I just do my zigzag. And then I put my loop in the inactivation. I make sure this is closed. I put it back in the baggie. And now I can put this in the fridge. So I can reuse it just in case my experiment, this doesn't grow, then I have a backup. When you put these in the fridge, because they've been open, they're no longer, sterile. Something from the environment could be growing in there. So if you are going to be reusing them, just make sure to check. And if there's something fuzzy growing or black growing, then that's mold or it's a environmental contamination. So just in that case, if you see anything that's not your bacteria that you know, just throw it in the inactivation bag and inactivate it. If you don't have the inactivation bag, then just fill a Ziploc or a container at home with bleach, a mixture of bleach water, and then we have a video for inactivation as well. So here we go. So now my Petri dish is ready. I can't see anything on it, right? Because the bacteria don't show up just yet. They need to grow first. But I know that I did touch the agar. If I look in the light, in the right light, right now I can see some streaks. I don't know if you can see it on camera. And so what I want to do now is actually incubate it, and I'm going to incubate this painting palette for 24 hours at 37. If you're using a homemade incubator, try not to go over 37. You can really even set it to like 35. That's fine, 30, 35. If you're doing it in the room temperature, just you're going to start to see it grow. It takes longer at room temperature, so it might take you 48 hours until you start to see colors. And you kind of want to keep it in a Ziploc bag or a plastic bag so that it doesn't dry out. For the same reason, I'm going to put this chamber, the humidity chamber, on it. I'm going to put this here, and then put it in my incubator. I'm going to lock it to make sure that the door is closed and that no one else can get to my Petri dish. And then I'm going to start the timer. So on screen, I'm just going to start the timer so that I know how long it's been there. Because I opened it, it went down, but 36 is fine anyways. And then I wait for that one to grow. For the rest of my experiment, what I'm going to do is I'm going to close these Petri dishes and I'm going to put them back in my baggie. All right, and this is going to go inside the fridge along with this. So what I can do is actually I can put everything here inside my baggie, everything I'll need for tomorrow, and put this whole thing in the fridge. Because it's open, if you're using a fridge that you also have food in, I would recommend putting this in a big Tupperware or plastic containers just to make sure it's separate from your food and that you're actually, any food organism don't get onto your Petri dish. I actually forgot to mention something when I did my Petri dish. So you might have noticed that I flipped it upside down, right? So I did that by habit, but I want to talk to you about that. So when I string my bacteria, it was the right side up, the agar was on the bottom. When I incubate it, I want to flip it upside down so that the side with the agar is facing upwards. And that's because I want to keep the humidity inside my Petri dish, right? I don't want the agar to dry out, and because of the way hair rises, this is the best way to incubate it. So as you can see, agar on top, lid on the bottom, then I put my humidity chamber, then I put it on the paddle, and I incubate. If you're doing this at home, if you're doing this you know, in a Ziploc baggie that you're leaving at room temp, also do flip it upside down so that the humidity can stay inside your Petri dish. This is really, you know, the one thing you need to watch out for now. You want to make sure your agar doesn't dry out. So now I have a few things. So I have some garbage from earlier, so these can go and be recycled. This can be recycled in the paper recycling at home or school. You can take off the staples so no one gets hurt. This can go in the garbage. These you can, if you want, try and peel off so that you can recycle the paper backing. And then just because it's mixed, it will go in the garbage. And I will get rid of those when the video is over. Again, this is going to go in the refrigerator, so I'm going to put that after the video as well. And then I have my bottle with agar, right? So I had a little bit left over. So what I want to do is I can get rid of that agar, and I liberate it from the bottom here by tapping it on a solid surface or tapping it with my hand. And what I do with this agar, this little surface of agar, is I can put it inside my inactivation bag. You might need to fight with it a little bit to get it out. Alright, you can also take a loop and try and scoop it out. Easier said than done, right? But why I want to do this is actually I want to reuse my bottle, because this is how we will put the bleach in. It really doesn't want to get out. So what I'm going to do instead is just leave it upside down like this, and eventually it will fall off. I don't want to fight with it. I don't want to spray agar everywhere. And so until tomorrow, what I can do is I can unfold this and zip it up so that all my materials are contained. Right, and then I can just leave this on my work surface because I know no one's going to come here and touch anything. And that's it. So I'm going to move this out of the way. I'm going to move my garbage out of the way and my recycling, and I'm just going to wipe down my surface. So again, I'm going to use my 70% ISO, spray my towel, and just wipe down my surface. This way, if I got any bacteria, if I got any agar on here, it's all clean and gone. I can even wipe the surface of my book here because it's plasticky. Alright, so now I know everything is clean. I can just throw in my garbage, put the kit in the fridge, and because I'm going to be reusing these gloves tomorrow, I'm actually going to spray a little bit of ISO on them, right? Just to decontaminate them. Okay, so now that they're decontaminated, I can take them off inside out. And I can leave them for tomorrow. We do have a video that shows you how to reuse your gloves safely. So as long as your gloves haven't actually touched the bacteria, you didn't slam your fingers in that tube or inadvertently touch something, you're fine to reuse them. If you did touch bacteria with them, then you should probably get a new pair out and put the gloves that have been contaminated with the bacteria inside your inactivation bag. But that's it for today. So tomorrow in 24 hours, we will start the day 2 of the canvas kit, which is all about making our art. This is now day 2 of the canvas kit experiment. We grew our painting palette in the incubator, so we're going to check on it, and if it's ready, we're going to start our painting. So I'm going to put on my gloves before I open the incubator. So it's just going to take me a few seconds. And again, we do have that video about how to reuse gloves if you want to learn about that, and that will be linked below. So I am ready. Because these are gloves that I wore yesterday, I am going to spray them with ISO. And you'll notice that I got the materials from yesterday from my kits, and I took them out of the refrigerator. I actually did tape them shut just to make sure everything was safe. So let's go to our incubator. So you can see it's actually been 28 hours, 42 minutes since I put in the painting palette in the incubator, and it's not because it wasn't ready, it's just I wasn't ready to continue the video. So the good thing about the canvas kit is that you can wait a little bit longer. It doesn't need to be a specific time period as long as you give your bacteria at least 16 hours to grow. So let's check on it. So I'll remove the humidity chamber and let's have a look. I have my blacklight flashlight, so I can see that the cyan grew very well. This is the magenta and the purple. There's a little bit of water in the top, so that's humidity, water that came out of the agar because I left it a little bit long in the incubator, so that's okay. I'm just going to be careful when I open it just to not make a mess. Here we go. Gently, gently. And you can see, yeah, my colors are there. Maybe it's hard to see on the video if I use this as a background. So I have pink, magenta, I have purple, and I have that cyan. So I'm ready to paint. So what I'll do is I'll put my painting palette here and I'll grab my stencils. I'll grab my sterile paintbrushes and I will grab my Petri dishes. What I'll also do is I'll open up my inactivation bag. So you'll remember from the previous part, I had closed it overnight. So now I've opened it up. I'm just going to spray my hands again since I touched the inactivation bag. And so what we're doing today is we're going to make our paintings. So because I'm using the home version of the DNA Playground, I only have place to incubate 2 Petri dish. So what I'm going to do is I'm actually going to paint only 2 pictures, and I'll keep my other Petri dish for the day after. So I'll put one back into the little baggie. And the reason I do this is that if it's my first time doing art, I want to see how the paint techniques, so when I'm using my sterile paintbrush, how they translate onto a Petri dish, because this isn't the same as using a coloring pencil. It's a new medium, so I want to see what my results are, and then I'm keeping one extra canvas that I can use later on. If you want to, you can draw all 3 canvases today, and if you only have place to incubate 2, you can put a pre-painted canvas back in the baggie in the refrigerator, and then once you have space in your incubator, then you can swap it in. So it can go into the refrigerator even if it's painted on but not incubated. I'm also going to grab my crayons that I'm going to use to paint. That I'm going to use to not paint, to draw my stencils with. So I'll be using the image stencil that comes in the kit. So I have the rocket ship, so I'm going to be using that for one of them. And then for the other one, I'm going to draw something. So the reason I have coloring pencils here is actually to mimic the color of my paint. So I'm going to use this one for the purple, this one for the pink, and this one for the cyan. If you want, we have another video that's tips and tricks about painting, so it goes into all the details of what to look out for and you know, different techniques for painting with the bacteria using the sterile paintbrushes. So I'm only going to need one of these, so I'll put these back in my bag because I don't need them right now. And I am going to I'm going to come up with something that I want to draw on my agar. So let's see, I'm going to use this crayon to mimic the purple, like I said. So my outline will be in purple. So I'm just going to go ahead. That's not very centered, but that's okay. And then I want to do kind of a thick outline because I'm going to be using a Q-tip for this, and you'll notice that the ends of the Q-tips are pretty bulky. So I want to make sure that I foresee what's going to happen. So I'm actually going to be drawing a kitty cat. Okay, so this will be the outline of my kitty cat, and it will be purple. I'll do a little, I guess that's its little leg. Here we go, a little paw. I guess it has 2 legs, right? Well, 4 in theory. Okay, this is not a contest right now, so I'm gonna continue with this, and then I'm gonna make its little nose pink. So this will be the magenta, little triangular nose. I'm gonna make its whiskers pink, and its little cat mouth. And then I'm gonna make its eyes cyan. Okay, so this will be our cat. Let's see how it turns out once we paint it with bacteria, right? Actually, I forgot the tail, little cat tail, so I'm just gonna do, should be rounded, little cat tail. All right, we're ready to go. So I have my painting palette. If I was painting with some fellow friends, we would all share the paintbrushes and the painting palettes, so that's something to be aware of. So what you do when you paint your pictures, you just put your Petri dish with the agar at the bottom on top of your stencil, and you'll see that they match up the size. So I'll just go ahead and prepare these. I'll open up my paintbrushes. So because I will be painting tomorrow, I'm gonna try and not use up all of my paintbrushes today. So I'm gonna be using some purple. Oops. Some purple. I'm going to be using some cyan. Actually, with cyan, I'm going to be using this for the pink. And then the cyan I'm actually going to do with the toothpick because it's only the eyes and little details. Oops. Come on. Okay. And you'll see I laid them out like on my painting palette so I can reuse them and always use the same paintbrush for each. So let's get started. So I'm gonna dab in. So you can see there's some bacteria on here. You don't actually need to see it. You just need to dab in, and you can see from the painting palette, you can see where I actually dabbed in. So I know I'm in the paint. So now I'm just gonna trace it. So you'll notice I opened up the lid, right? And now I'm just gonna trace. And I want to remember that the agar is soft like jello. So I don't want to press too hard. So right now I'm just tracing the magenta with my magenta. I'm also going to do the little fire. You'll notice that I don't need to go back in and get more paint. That's because there's actually thousands, billions of bacteria on the Q-tip already, so I don't actually need to go back and dab in. They're already there. Then I'm going to take my purple, just dab it in a little bit, and then I'm going to do my purple, I guess those are the rocket ship fins. Right, here we go. Going to do the one in the middle, and I'm going to move my paintbrush back to the Petri dish. Then I'm going to grab with the toothpicks, I'm going to grab some paint on it. I can not see it, but because I was careful when I dabbed it in and I saw that there was paint here, and I took it on my toothpick. You can also make sure you shine your light so you can see you're definitely touching some. And actually you can see it now. Alright, and now with the toothpick it's a different technique, so it's actually going to make lines in the agar because it's pointy. So I'm just going to go and trace this. And usually with the toothpick what I like to do is just little dots because it's easier than just making straight lines. I'm going to do my stars. And of course if you're using a pre-made stencil you can feel free to add details as you want. I added a few more stars. And I'm going to lift it up and I can see in the light, so I can't see any bacteria on here, and that's the beauty of painting with bacteria. It's like an invisible ink, but in the light I can sort of see some wet traces, right, where I did drag either my toothpick or my Q-tips. So I'm happy with this. What I'm going to do is I'm going to put the lid back on, and I'm going to wait until I'm done my second painting, and then we'll incubate this. and I'll put this stencil back in the bag. All right, kitty cat time. So again, I'm going to use the same paintbrushes. You can reuse them as often as you like during the same day. And then I'm going to open up the lid. And again, I'm just going to try and trace my image. So I'm going to dab again just to make sure I have some paint. The darker pink here in this image is supposed to be my purple, so I'm just going to trace it. I want to be very gentle because the agar is soft. If I were to inadvertently lift my Petri dish, then one way I can try and put it back to where it was is by looking at the shadow. So I can see a shadow of where I've traced it. I don't know if that's obvious on camera, but you'll see when you make your own art that you can see wet traces where you did put the bacteria. Okay, so I just did the tail. Now I'm going to do the nose. So that's going to be in pink. And then I'm going to do the little cat mouth area. I have a feeling that's not going to come out too clear, simply because I ended up using the Q-tip, and that's a really large end. I should have maybe used the toothpick. So what I'm going to do is I'm going to swivel this around, and I'm going to go collect some magenta paint. And now I'm going to do my whiskers with the toothpick, and they're a little crooked. And that's okay. And that's really also a reason why you would want to maybe keep one or two canvases for after your first try, because this is not as easy as it looks, because you can't really see what you're doing, and the jelliness of the agar makes it a little bit more tricky. So while I was speaking, I went back in, I swiveled my toothpick around back to the cyan, and I collected some bacteria, and now I'm making the cat eyes. Because this toothpick has bacteria on both sides and I'm done with my painting, I'm just going to go ahead and put it in the inactivation bag right away. Alright, so this is my cat painting. You can sort of see an outline. You can definitely see where the toothpick was. We'll see. I am maybe 60% confident that this will turn into a cat, so that's something we'll have to wait and see. So what I'm going to do is I just put the lid back on, I'm flipping it, and I'm preparing it to put it back in the incubator. Because I am done with my painting for today, I'm going to seal my little paintbrush bag back up, because I still have one canvas, so I still have all these painting instruments. I can also use some of these loop to paint with. I decided not to because I'm more comfortable with the Q-tips and toothpicks, but you can try a few different things out, figure out what works for you. And then I'm going to take the paintbrushes that I did use up, I'm going to put them in my inactivation bag. I'm going to put my stencil, I'm going to put it back in the kit. I kind of want to see how the painting compares to what I wanted to achieve. This extra Petri dish also goes back in. We'll put that in the fridge. I will move these away. And then I have some, like I mentioned earlier, I have some water inside my lid. So what I'm going to do is I just tore off a piece of paper towel, and I just absorbed it with the paper towel that I will now put inside my inactivation bag. Right, because there might have been bacteria there. So that goes in the inactivation bag, and then I'm able to put my lid back on. The reason I absorbed the water is just because I didn't want to make a mess when I closed it back up. You could, if you didn't have that much water, you know, you could actually close it up, and that's fine. So I'm going to go ahead, put my humidity chamber on top of my two plates, and those are upside down. Agar up, right? Just like yesterday. I'm going to open my incubator, and I'm going to put the stack in. I'm going to lock it up, and I'm going to actually reset the countdown so that I know how long they've been incubating. But here we go. So they're still at 37. If I don't have an incubator, again, I can do this at room temperature. If I have a home or like a classic life science incubator, I can use that, just set it to 37, that's no problem. And what I'm going to do with this painting palette, because I will need to use it tomorrow, I'm actually just going to put it with my extra canvas. So I'm going to put it inside this plastic bag. Again, the reason we put agar in plastic bags for most of everything is just to prevent the agar from drying out. So I'm going to seal the bag, and this is going to go in the fridge, and I can leave my painting palette in the fridge for a couple weeks as long as I don't see any mold or weird, you know, fuzzy growth or anything growing that's not my colors. I can keep it in the fridge and use it later on. For a couple of weeks, your bacteria will still be alive for you to use. So now I'm just going to close my kit back up. I'm going to use that piece of tape that I had and just fold it up, just seal it, just to make sure that all my experiment material stays together. And I'm going to put that in the fridge. I am done for today, so I'm just going to clean up my space, because you saw that my paint brushes, some of them went on the table before after I dipped in bacteria, which means there's bacteria on my table now. So I'm just going to wipe it up. And again, I'm using this sort of metallic table for this specific reason. It's not porous, so nothing's going to get inside the table, so I can clean it up without problem. I might have gotten bacteria on my gloves, so I'm going to wipe them with ISO as well, but I'm also going to take them off. And I'm going to throw them in the inactivation. The reason I'm putting the gloves in the inactivation is because I think I touched some bacteria, so these should be inactivated. If you didn't touch any bacteria, if you didn't drop your paintbrush or anything like that, you don't need to put your gloves in the inactivation. You can either rewear them or throw them out in the regular garbage. But that's it for today. So tomorrow in 24 hours, we're going to check on our painting, and then we're going to do our last painting using what we've learned, right, from the results. And so for now, I'm just going to, again, I'm going to close my inactivation bag. I don't need to do anything with it right now. I'm just going to wait till the end of my experiment. I'm going to leave my incubation running. I can start checking on my art 12 to 16 hours after I did it. I'm going to start seeing some faint white appear, and the bacteria are going to turn white before they turn color. So that's something to note. And yeah, if I plan on if I was going to incubate them for longer than 24 hours, say I didn't have time to do my other one, or I thought the colors were a little bit faint, what I would do after 24 hours is actually change the temperature to go to 30, right? So I would want to incubate them at a lower temperature. And if I incubate them at a lower temperature, so 37 for the first 24 hours and then 30 for the next 24, is going to prevent that agar from drying out. If I'm in the classroom and it's a Friday and I just want to leave them in the incubator until Monday, I can put it at 30 instead of 37. So if you need to do that, if you want to wait for longer than 24 hours before you see your result, leave it to 30, because that way you're not going to get anything overgrown. The bacteria aren't going to take over the entire Petri dish, and you're also not going to dry out the agar, but the bacteria will still grow. All right, we'll see you when the paint is ready. Welcome back. This is now day 3 of the canvas kit. What we're going to do today is we're going to look at our paintings that have been incubating, and we're going to do our final one based on what we learn from our art. So I took my bag from the refrigerator. I'm just opening it up. What I'm going to need today is my painting palette, my last Petri dish, my paintbrushes, so my sterile paintbrushes, and some stencils. I'm not sure if I'm going to retrace something I already traced or do something new. Let's look at the art to find out. So they've now been incubating for 27.5 hours. You can see that on the playground. So let's just see what they look like. All right. Ooh. So let's put them next to our stencil. It looks like the kitty cat's actually not bad. It's got some nice glowing eyes. Let's see if I can open it up. I'll put it on top of this so you can see him. So the eyes only show up under UV, but he's pretty cute. You can definitely tell that where we did the nose and the cat mouth area, it's a little thick. It might have been better if I had used the toothpicks for this, and there are little dots here and there, right? But overall, it's pretty good. And then again, ooh, the eyes shine under UV. So, good job on the kitty cat. I will leave it inside a baggie at room temperatures because the color may get a little bit darker. The purple might get a little bit darker. And then let's look at the rocket ship. So, the rocket ship is okay. You can tell that my agar must have been wet because there's a big smear of cyan bacteria. Let's see if you can see that all the way along here, and that wasn't something I had planned on it. You'll remember I was just doing the dots for the stars. So it must have been wet, and so the wetness kind of mixed the water on the plate, mixed in with the bacteria and made these long lines. It's not terrible, right? But it's not what we expected. So that is agar art for you. So from this, we learned that maybe the toothpick is a little bit better to do details like the cat nose area. To be careful that our agar is dry, otherwise you do get those streams, but actually it looks a little bit like flames. So you could use that in your painting if you wanted to. So today we're going to do our final painting. I'm going to check. The agar looks pretty dry, so I shouldn't have any of those streaks. And then I've got my painting palette right here. So I've got my painting tools. I'll just lay them out like this. And I will grab my drawing tools and I'm going to draw one last stencil. So when it comes to it, you remember that we were going to use this color for the purple paint, this color to signify the cyan paint, and this color to signify the magenta. And really we saw that fine details are okay if you use the right painting tool, and that really, you know, you can pretty much paint whatever you want as long as you take the time to actually apply yourself and transfer it into bacteria. So what I'm going to paint this time around, I'm going to try and do a little bacteria like the one we have here. So I'm actually going to do the outline in purple. Like this. And then I'll do a little flagella. I'll do its superhero mask in pink. Right, so that'll be a nice line size that we can do with the Q-tips. I'll do, actually, you know what? I'm going to do yeah, that's a better idea. I'm going to use cyan to do the mask. That way, it's only going to appear under UV. And I'm going to do the same for the cape, a little superhero cape. And then I'll do the eyes in purple as well. All right, here we go. And it looks like I'm not necessarily going to be using the magenta. Oh, I'll do the smile in magenta. Why not? Here we go. So now it's just like last time. I'm placing my Petri dish on top of my stencil, and I'm just going to use my painting palette and my painting instruments to draw. So I'm going to do my outline first in purple. Remember, you don't need to collect so much paint. You don't need to do a big swab. This is already plenty. I'm going to do little dots. Oops. And I'm just going to do an outline. I don't think I'm going to color it in. Little flagella. Going to do the eyes. Actually, with the eyes, I'm just going to do a dot here. One eye, two eyes. And I'm done with the purple, so I'm actually going to put my paintbrush in my Inactivation bag. This is my last painting, so I don't need to hold on to them. I'm going to use the toothpick to do a magenta smile. And I don't know if you can see, but I'm choosing to do little dots. I just find that that's a good way to apply the paint. So that's it for this painting instrument as well, Inactivation bag. And last but not least, the cape. So you can see again, there is some cyan there, right? Alright, so I kind of moved this around. I don't know how it's going to turn out, but let's do the mask. Now I'm going to try and not go over my eyes because that could wipe away the color, so I need to be careful. So I'm actually going to lift it up to the light. My last streak. And then I want to do the cape. So here I'm realigning it. I'm going to swap the side of my Q-tips just because I think I might have gotten purple on here and I don't want to smear it around. Alright, let's try and do the cape. So I'm not sure how it's going to turn out, but we'll see. It looks decent. I'll put it in the incubator and we'll see in 24 hours what it looks like. So to do that, I'm going to put the lid on. I'm going to flip it upside down, right? Put my humidity chamber, and I'm just going to put it in, lock that up, reset my timer. And so now that's incubating. These two, I think I will keep them. I haven't taken photos or anything of them just yet, and I think I could get maybe a brighter color if I leave them at room temperature. So what I'm going to do is I'm actually going to put them in my plastic bag so that they don't dry out. So that's perfect. I have one toothpick left over, so in theory, if I had more Petri dish to do at home, or if I knew I was going to do this again, I could keep it. So I'm just going to put that in. So now I'm done with my experiment, really, except for getting the new painting, incubating, and keeping these and taking any photos that I want. So what I can do is actually take all of my materials. So I'm done with my bacteria. I don't need to hold on to them if I'm done with my painting. Same thing with my painting palette. So I'm actually going to put these in the Inactivation bag. We have another video that shows you how to inactivate. So we'll put the link up so that you can watch that video. And then for the rest, I am going to hold on to my stencil so that we can compare our art when this one's ready. I am going to keep these loops that I didn't use because they're sterile, and if I were to do another experiment, I would have these in the future. I am going to keep this toothpick as well. I can now start cleaning up my space. I can put away my material that I've used. Anything that has not touched bacteria can just go back on your shelf. And then these I'm keeping at room temperature for a couple more days. If I'm done with them, or if I want to show them, for example, to my student in a couple weeks, what I can do is actually put these in the Ziploc and put them in the fridge once I think my colors are bright enough. I'll keep them in the fridge for a couple of weeks until I can show people, you know, what the results were. And after that, I would inactivate them. I would take photos, make videos if I want, and then inactivate them. The other thing I can do is actually use something called the Keep It Kit that we offer, and that's a special resin that you can actually pour on top of your Petri dish to keep them going for a long time, so that you can share them with your friends, bring them home if you did these at an event. But we'll still be back tomorrow, so I can show you what that looks like tomorrow. And right now I'm going to put these right next to my playground so that they keep going. I can check on them tomorrow. And I'm going to put this material inside my science experiment, you know, container where I keep all my science experiment safe. And that's it. So I'll see you in 24 hours. Hi everyone, welcome to the last day of the Canvas kit. What we're doing today is we're looking at our last painting, and that's pretty much it. We have a separate video for inactivation, so you can follow that when you're ready to inactivate all your materials. And then we also have a video about the Keep It Kit, and I mentioned that in just the earlier segment that you can keep your painting by pouring resin on top from the Keep It Kit. And what that actually does is it creates a resin surface on top of your Petri dish. So I have these Petri dish that have been resin. So you can see it's like a very hard surface, right? And it's on top of my bacteria, so that means my bacteria is actually protected, it's encased in the resin, so it means I can actually touch it like this and I can keep them. So you can also see the UV does shine through it. So that's how the Keep It Kit works, and it also comes with a little stand so you can put these on display. So if we go back to our art, so we have our art from last time. They were in my plastic bag. I just opened it. And they've continued to get darker in color. And what we're actually going to do is we're going to look at them in the black light box. So while we do have this little flashlight, it's nice to see. The UV shows much better if it's encased in a black environment, so when there's no light present. So we're going to look at them in the black light box. And we do have a blog post on our website that shows you how to make this black light box. But as you can see, pretty simple. You kind of just want a black background to your box. You can use an old shoebox, an old shipping container, and then you put your black light in it. We use this UV light, this UV black light for this purpose. And without further ado, let's look at our last painting. So I did have a peek at him earlier. He's an interesting bacteria, but basically this is him. So you can see he does look like a bacteria, and it does glow. I would say that maybe the cape here was a little bit unclear what's going on this side, but overall a valiant effort. So let's put him in the black light box and see what that looks like. Just gonna turn it on. Alright, so you can see now that this is only the cyan and the magenta are glowing. I don't think there was much magenta in this painting actually, so it's pretty much a cyan picture. Let's continue on with looking at the rocket ship. Again, this one we have those, streaks, and they're from the fact that actually our agar was wet, so those created streaks, but they kind of work with the space design, right? So that's what this one looks like under black light. Again, the purple does not show under black light, so that's why you can't see it. That's what it looks like under regular light. And finally, our little kitty cat friend. Gonna put him under. Okay, so right now you can only see pretty much its eyes, which is very much what would happen if you looked at a cat in the dark. So a good success, I'm gonna say. And you can see some of the whiskers and some of the ear details from where the magenta is. I would say that our cat is our best result for this art. And if I was going to keep any of these, I would resin the little kitty cat. So there you go. That's agar R bacteria painting with the canvas kit. You can see our successes. Some would benefit from a little bit more work, but that's what it's all about. So if you have any good paintings, please share them with us. And we also have those other videos I mentioned. So inactivation, which is what we would do now. We would put all our materials in this inactivation bag just to make sure that everything is clean, that we're disposing of everything in a safe and responsible manner, because that's really important when you do science. And we would put all our paintings in here, add bleach water, let it sit for 24 to 48 hours, and then discard of the liquid in the toilet or the sink, and the rest can go in the regular garbage. There you go.